My name is Erin Jennings. I'm an undergraduate junior food science major at the University of Delaware. With the collaboration of Carmen Smith and supervision of Alexis Omar and Camilla E. Neal, I researched the persistence and transfer of E. coli 0157H12, a study for produce safety. This study was performed to investigate the concentration of Escherichia coli 0157H12 in cilantro plants over time with varying water activity. E. coli 0157H12 is a non-pathogenic E. coli strand, but can provide insight on how pathogenic strands of E. coli may act in similar conditions. Cilantro is planted in a mix of soil and poultry litter. Samples were kept at temperatures between 19.8 and 25.6 degrees Celsius and humidities between 52.1 and 81.7%. The soil of the samples started at a water activity of 0.983. The water sample ended at a water activity of 0.937, while the non-water sample ended at a water activity of 0.603. To set up the experiment, soil and poultry litter was measured and mixed. The soil mix was then portioned into 30-gram sampling cups. Cilantro plants were planted into each sampling cup. 1 milliliter of 1 times PBS was added by drop inoculation into the soil of water and no water samples. Samples were set on a window ledge until sampling occurred. For sampling, the cilantro is separated from the soil using tweezers. The plant and soil are then placed into Whirlpack bags. Tryptic soy broth was added to all bags to make the sample easier to work with. The bags were hand massaged for 1 minute each to mix thoroughly. The picture shows before and after mixing, left and right respectively. Samples were then diluted by doing a series of 1 to 10 dilutions. Diluted samples were plated onto McConkey agar, supplemented with nalidixic acid. Plated samples were incubated overnight and then counted and recorded the following day. Each sample type was graphed to compare the log of colony-forming units per milliliter and sampling day. E. coli concentration was consistently higher in samples that did not receive water treatment. This was seen in both soil and cilantro samples. Samples generally followed a standard bacterial growth curve, with cilantro no water showing the most deviation from this model. Deviation was most likely due to the small sampling size and limitations of disruptive sampling. All samples started to see a decline in E. coli concentration around day 5. More samples are needed to determine if a correlation between water amounts and E. coli concentration exists.